Hi, everybody. Welcome or welcome back. Today, we're going to be explaining cell signaling and cell respiration. I'm your presenter, Kim. So first, cell signaling. Cell signaling is how cells communicate with each other. It can be via electrical signals or chemical hormones. So here is a pathway showing how a signal from outside is transmitted throughout the cell and amplified. So first, the ligand, a chemical, binds to its receptor, and it is specific and complementary to that ligand. This triggers G protein to activate an enzyme, and that enzyme makes secondary messengers. And these secondary messengers start an enzyme cascade. For example, if this was a insulin receptor, then insulin would bind, and only liver and muscle cells would have the insulin receptor because one of their functions is to store fat. Any other cell which don't have the function of storing fat will not have an insulin receptor. So here is the pathway of glucagon. So glucagon binds to its receptor. G protein is activated. It activates adenyl cyclase, which catalyzes the reaction of ATP to CAMP. CAMP is the secondary messenger, and it starts a enzyme cascade. It activates protein kinase, which activates phosphorylase kinase, which activates glycogen phosphorylase. So notice how everything has ASE at the end, meaning they're all enzymes. And anything that has kinase in its name means that it activates all the molecules by phosphorylating that molecule. So the receptors in the illustration are cell membrane receptors. They are intracellular receptors as well, meaning these receptors are inside the cytoplasm, and the ligand has to cross the phospholipid bilayer to get to the receptor. In that case, the ligand is small enough to diffuse through the cell surface membrane, and is hydrophobic. After the ligand receptor complex forms, the complex can then move to nucleus and regulate transcription of a gene, for example, with estrogen. Estrogen binding to its receptor. After estrogen binds to its receptor, the estrogen and receptor complex move towards the DNA to the nucleus where it's going to affect gene transcription involving transcription factors. So that is one of the possible responses for cell signaling. Other possible responses are secretion of chemicals, which is known as exocytosis, transcription, movement, and metabolic change. The next is cell respiration. There are four stages to cell respiration: glycolysis, Link's reaction, Krebs cycle, and oxidative phosphorylation. Cell respiration is the controlled release of energy from organic molecules. The energy is then captured and used by the cell in the form of ATP. So first in glycolysis, the glucose is phosphorylated to make it more reactive. The molecule becomes unstable and breaks down to produce two pyruvate molecules. NADH, which is a coenzyme, it carries energetic hydrogen atoms. So when the molecule breaks down, it releases energetic hydrogen molecules, and the NADH picks them up and acts as a kind of like a taxi. At the same time, because of how unstable it became, ATP is synthesized via substrate-linked reaction. Next, the pyruvate moves into the mitochondria when oxygen is available because it is co-transported to take part in the Link's reaction. So in the Link's reaction, pyruvate is metabolized to produce acetyl coenzyme and reduce NADH, and carbon dioxide, which is given off from the cell. Acetyl coenzyme enters Krebs cycle, and it provides the acetyl group needed to regenerate citrate. The citrate is decarboxylated and dehydrogenated in a series of steps. Carbon dioxide, ATP, NADH, and FADH. Two are produced. So FADH, like NADH, is a coenzyme. It is another carrier of energetic hydrogen atoms. So the carriers NADH and FADH, they move to the electron transport chain to drop off their hydrogen atoms. After being dropped off, the atoms split into protons and electrons. The electrons are passed around to use the energy to actively transport protons into intermembrane space, which is this pale purplish space here, which creates a proton gradient. So the protons diffuse down their concentration gradient through ATP synthase, and energy released from that movement is used to synthesize ATP. So this is the pathway we just saw for how glucose is metabolized in normal cells to produce ATP. We can see that first in the cytoplasm, glycolysis takes place and it moves into the mitochondria, and then Lynch reaction, Krebs cycle, and oxidative phosphorylation takes place. A few other elements are involved, such as amino acids and fatty acids. But in the case of a proliferating cell, a cell that is rapidly dividing, the pathway is not so straightforward. Yes, ATP is still produced, substrate-linked reaction, chemiosmosis, oxidative phosphorylation still occur, but glucose is now used to form nucleic acids, to form proteins, to form lipids, which are all essential molecules in a cell that is dividing for nucleic acids, DNA replication, transcription, proteins, kinases, lipids. A proliferating cell would also consume more glucose. It would use it up a lot faster. It would use it in a lot more processes. So we have sort of covered the basics, explaining the processes behind hallmarks of cancer. So again, sustaining proliferative signaling, evading growth suppressors, resisting cell death, inducing angiogenesis, activating invasion and metastasis, 1, 2, 3, 5, 6, and 8, evading immune destruction. They're all involved with cell signaling. Most oncogenes, they code for growth factor receptors. So that would explain the proliferative signaling. So cancer cells make it so that they're always receiving signals to divide. Some oncogenes such as SRC and BCR-ALB. So BCR-ALB are two different genes, but because of a indication, those genes have linked code for a type of protein called a tyrosine kinase. Again, with a kinase, kinase is an enzyme that phosphorylates other enzymes, that activates other enzymes, and be like, oh my gosh, everyone, get up, do your job, especially if the kinase is part of the pathway of cell division. If that kinase is always active, cell division will always be active. So the constant production of the kinase, the constant transcription of the oncogene will lead to cell proliferation. And your reprogramming of energy metabolism is what I just said previously, how the cell uses glucose, how it metabolizes glucose is altered so that it can produce other essential biological molecules as well. And yeah, that's all. And thank you so much for watching and supporting me on the series. Until next time.